Good morning. It's May 9th, and this is your daily brief in science. Here's everything you need to know. An innovative interactive map named MiCO has emerged from a collaboration between the University of Queensland and Duke University. This tool illustrates the migratory connections of over 100 marine species, providing valuable insights into their patterns and behaviors. MiCO, published on May 8, 2025, in the journal Nature Communications, compiles thousands of records from more than 1,300 sources covering 109 species, including birds, mammals, turtles, and fish. The research spans marine migratory patterns from 1990 to 2017, underscoring the critical role these migrations play in maintaining ecosystem health. The findings reveal how animal migrations connect diverse habitats and ecosystems worldwide, which is essential for the well-being of both migratory species and their environments. MiCO also highlights the need for cross-boundary cooperation, as many migratory animals traverse national waters and high seas, facing various threats along the way. Migratory paths often cross multiple jurisdictions, necessitating international collaboration for effective conservation. This is particularly relevant in regions like longline fisheries, which pose risks to species such as albatross. Associate Professor Daniel Dunn noted that the connectivity data from MiCO supports global conservation initiatives, including the High Seas Treaty, aimed at preserving biodiversity beyond national waters. Furthermore, MiCO's models serve as valuable resources for informing policymakers about their country's relationships with high seas biodiversity and for implementing effective protection strategies. Despite the extensive data available, researchers indicate that more than 2/3 of marine migratory species remain unassessed. Future expansions of MiCO are planned to enhance data comprehensiveness for conservation strategies. Dr. Lily Bentley emphasized that protecting migratory species requires collaborative efforts among nations, as no single country can achieve this alone. The interactive map is accessible at https://mico.eco/, offering a user-friendly platform to explore specific species and their migratory connections. Recent research from UVA Health has revealed that nucleoside reverse transcriptase inhibitors, or NRTIs, commonly used in HIV treatment, may significantly lower the risk of developing Alzheimer's disease. This study analyzed data from over 270,000 patients aged 50 and older, excluding those with prior Alzheimer's diagnoses, across various health databases. The findings were published in Alzheimer's and Dementia, the Journal of the Alzheimer's Association, and received support from grants from organizations such as the National Institutes of Health. One of the strengths of this study is its large, demographically diverse population, which helps to ensure that socioeconomic status does not confound the observed association between NRTIS and Alzheimer's risk. However, the study does have limitations, including its reliance on administrative claims data and medical coding for Alzheimer's diagnoses, instead of clinical assessments. Current statistics indicate that nearly 7 million Americans are affected by Alzheimer's, with projections suggesting that this number could rise to 13 million by the year 2050, leading to escalating care costs. The researchers propose that NRTIS could be utilized more broadly for Alzheimer's prevention, extending their use beyond just individuals at high genetic risk. To address toxicity concerns associated with long-term NRTI use, the research team is developing a new drug called K9, which aims to be safer and more effective. K9 is currently undergoing clinical trials and seeks to mitigate adverse effects linked to traditional NRTIS while potentially offering benefits for Alzheimer's disease. In addition to NRTIS, the team is advocating for clinical trials of K9, which is already being tested for other diseases. Previous studies have noted a reduced risk of Alzheimer's in NRTI users, and this research employs a more rigorous methodology to validate those earlier findings. A recent study published in the journal Cell has unveiled a significant breakthrough in the field of gene regulation. Researchers from the Center for Genomic Regulation have developed an innovative AI tool capable of designing synthetic DNA molecules that can control gene expression in healthy mammalian cells. Over the course of 5 years, the team synthesized more than 64,000 synthetic enhancers, resulting in the largest library of synthetic enhancers specifically for blood cells. This extensive library was analyzed across various stages of blood cell development, providing a comprehensive understanding of gene activation requirements. To create the AI model, the researchers generated a substantial dataset through thousands of experiments focused on blood cell formation, using healthy cells to ensure an accurate biological representation. The AI model can predict combinations of DNA sequences necessary to achieve specific gene expression patterns, facilitating the creation of synthetic DNA fragments that activate genes in targeted cell types. In a proof-of-concept experiment, the researchers successfully designed DNA sequences that activated a gene coding for a fluorescent protein in mouse blood cells, demonstrating the model's capability without disrupting other gene functions. This advancement could significantly enhance gene therapy approaches by enabling targeted modifications to genes in specific cells, aiming to minimize side effects and improve treatment efficacy. This study marks a shift in generative biology, transitioning the focus from protein design to precise control over gene expression, which is essential for addressing diseases linked to faulty gene regulation. The findings also highlight the complexity of gene interactions, as many enhancers can activate genes in one cell type while repressing them in another, a phenomenon referred to as negative synergy. Dr. Robert Fromell, the lead author, noted the transformative potential of this technology for gene therapy, enabling precise adjustments to gene activity in targeted tissues. The AI-generated enhancers promise to enhance the precision of therapeutic strategies while minimizing unintended effects on healthy cells. Recent research from the University of California, San Diego has unveiled a groundbreaking insight into how learning physically rewires brain connections, enhancing communication between various brain regions. This study, led by Professor Takaki Komiyama, demonstrates that during the learning process, the thalamus activates specific motor cortex neurons while inhibiting others not involved in the new movement. This action refines the neural circuitry related to motor tasks, providing a clearer understanding of how our brains adapt to new skills. To deepen the understanding of these mechanisms, the research team developed an innovative data analysis method called SHARE-D, or Shared Representation Discovery. This method identifies common neural representations across different subjects, allowing researchers to combine data from multiple experiments. Such a technique facilitates discoveries that would be impossible with limited data from individual subjects, offering a more comprehensive view of neural activity associated with learned movements across various animals. This study builds upon earlier research from the Komiyama lab that explored synaptic mechanisms and neuron behavior during learning, further advancing knowledge of neural circuit development. Lead author Asaf Ramot emphasized that learning reshapes not only brain activity, but also the wiring of brain circuits, making them faster and more precise in performing tasks. This discovery is particularly significant as it provides concrete evidence on how brain cells interact during the learning process, filling a crucial gap in neuroscience. The study is dedicated to An Wu, a talented assistant project scientist in Komiyama's lab, who tragically passed away in a fire in 2023. Her significant contributions to neuroscience are honored through this research, underscoring the impact of her work in this vital field. A recent discovery has unveiled a concerning capability of the bacterium Pseudomonas aeruginosa, a pathogen commonly associated with hospital infections. Researchers have identified that this strain can produce an enzyme named PAP1, which is capable of degrading polycaprolactone, or PCL, a medical-grade plastic frequently used in healthcare settings. This finding represents the first known instance of a pathogen that can break down plastics typically utilized in medical devices, raising serious questions about their integrity. In laboratory experiments, exposure to PCL was shown to increase biofilm formation by Pseudomonas aeruginosa. This biofilm formation can enhance the bacterium's antibiotic resistance, making treatment efforts more complicated. The ability of this bacterium to degrade plastic fragments not only aids in biofilm development, but also provides protection from antibiotics, further complicating infection management. Notably, the strain Pseudomonas aeruginosa PA-W23 can utilize PCL as its sole carbon source for growth, suggesting it may thrive in hospital environments where such materials are present. This capability raises alarms about the potential for prolonged infections, particularly given that Pseudomonas aeruginosa is already a significant concern in hospitals due to its resistance to antibiotics and its association with severe infections, including catheter-related urinary tract infections and ventilator-associated pneumonia. Research has demonstrated the effectiveness of the PAP1 enzyme in breaking down PCL under controlled laboratory conditions. Published in the journal Cell Reports, this study challenges the previously held belief that pathogens cannot degrade medical plastics. The findings indicate a need for revised infection control practices and suggest that hospitals may need to consider using more durable plastics to mitigate these emerging risks. Experts are calling for a reevaluation of monitoring practices in hospitals and the exploration of plastics that are less digestible by microbes to enhance patient safety. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.